Hello everyone, my name is Alencar Xavier and I'm here today to talk to you a little bit about the technical nuances of machine learning in plant breeding. So you may have seen or heard somewhere that information is both power and profit. Yet I would add to it that the adequate use of information is both power and profit, as it takes some mindful use of data to extract the adequate information you need to do a good some good data-driven decision. So my outline today is just like the movie. I'll be talking about the good, the bad, and the ugly faces of machine learning, where good refers to the potential, the bad refers to the indiscriminate use and some of the biases, and the ugly correspond to some of the inner workings of machine learning. Alright, so let's get started with some introduction and motivation and the concept of filters in plant breeding, which I find it fascinating. So the solution is motivated by the problem. So what we have been witnessed in the past few years is the increase in the amount and quality of data. So we have more phenotypes as they have been collected by drones and high throughput technologies. We have more genotypes that have become cheaper over time and has motivated, as of course, higher density genotyping and the collection of the information and use for genomic prediction. Likewise, we have been observing that more and more robust environmental data has been made available to the public. Here are some examples of public databases from, including NASA, Harmonized Soil DB, the USDA, GridMet, pretty good, and the National Weather Service. And in addition to having all this amount of data, we have more computing power now as cloud services have become more inexpensive. So basically, having all the data in the computing makes all the ingredients to bake the cake. So what is machine learning good for after all? So I would say that machine learning works very well when you have one single well-defined problem. So in this picture I'm depicting on the left a robot that is climbing stairs and making pancakes. And on the right we have two robots, and where one robot is climbing stairs, the other one is making pancakes, so they are more specialized. So if I were to choose which of those pictures better depict a good use of machine learning, I would say that the one on the right, because what we're really looking for is to optimize from point A to point B and having a very simple and straightforward line to follow. So machine learning is and has been used in plant breeding for a while. So there are two major branches of machine learning, supervised and unsupervised learning. So supervised focus on problems that have a response variable, and you really want to do that modeling and prediction. Whereas unsupervised focus on capturing patterns in the data and associations. So unsupervised learning is used for clustering and associations when you want to do, germ plot classification and environmental classification. Whereas supervised learning is used for prediction per se. And the branch we are going to focus today is the branch of omic-based prediction, really. So supervised learning is really nice, and it can be interpreted from this perspective of statistical filtering. So a filter, as depicted here, is a prediction machine where you pass your data through and get some signal and some noise, which are more often than not perpendicular to each other. Filters can be anything from regressions or kernels or networks or tree-based filters, so all the machines can be used as filters. So from all the supervised machine learning methods we are accustomed to, four major methods of applying supervised learning: regression type, kernel type, which are two that we use a whole lot to generate breeding values, and signal detection. And there's also the neural network and tree, which as of right now have been primarily used for general purpose prediction, such as phenotypes predict phenotypes, phenomics, and general, as opposed to capitalizing on a specific type of breeding objective. So the idea of filter can be generalized. So as I presented earlier, filter would be something that you enter your response, your phenotype, and get your signal and your noise. It's also possible to imagine that you can apply simultaneously multiple filters. And the idea of applying multiple filters is conceptually conceivable when you have tangled signals and you want to untangle them. So when you apply multiple filters, what you get is your residuals or your noise and the two separate signals. And another type of filter is the multi-response filter, where multiple inputs get into the filter and you get a single corresponding signal and noise for each of those inputs. But why should we care about multiple filters and multi-response filters? So one example of multiple filters in plant breeding is when we are trying to separate or untangle field variation from family layout. So here's an example from the SOINM, where it's a field from 2014 in Indiana, where you can see that 4 families fell into this very unfortunate spot of the field that was at the time hammered by SDS. So in order to differentiate a good family that was placed in a bad spot and a bad family that was placed in a good spot, we apply 2 filters to it. So in this case, we are applying G corresponding to the genetic filter and R a spatial filter. Those could be a genomic relationship or pedigree, and R could be a AR1 by AR1 or a spline of a sort. So what we get out of those basically is that the spatial signature becomes very clear and hopefully untangled from the genetics, since we don't see trends that follow the family structure. On another hand, the genetics here show some of the blocks of families, but there's no clear side of the field that's better or worse. And the residuals should present no pattern. So that's the desirable depiction of residuals. So we made a simulation to see what's the gain you get from applying those filters concomitantly. And a very nice thing is that here we are looking at genetic accuracy and the spatial accuracy in the simulation. In left and right here basically is not using relationship and using genetic relationship, and the different types of filters for the spatial, so row column, AR1 by AR1, or spline. So of course, adding the genetics really gave a boost into the genetic accuracy, right? If you add the relationship information, could be a genomics or pedigree, in this case genomics. But adding the genetics and this relationship also increase, although not by much, the spatial accuracy. So that's a good example where you're getting an untangled, because you can clearly see that adding a relationship is giving you a more accurate spatial accuracy. Likewise, here the no spatial adjustment, you have less genetic signal than places where you have some spatial adjustment, as in AR1 by AR1 or A spline. So you get a lot of synergy from those multiple filters being applied together. And why should we bother, on the other hand, with multi-response filters, right? Because if we do not apply multi-response filters, what you get is a very parallelizable, and if you work on a big pipeline, that might be desirable. But on the other hand, if you do decide to use a multi-response filter, there's one piece of information you get out of it. When you apply multiple responses, you do not necessarily assume that the signal from 2 of those inputs are 0. Basically, if you are running, for instance, a multi-trait model, what you get is the additional information that comes from the genetic correlation. So that's the case where it's very common for a GBLUP, and you run a multi-trait GBLUP and you get those genetic correlations. So that's extra information that would make your machinery more accurate at no additional cost or unless there might be some computational cost involved. So the nice thing is that it can be mathematically proven that the multi-response filters add some gain. Here, of course, is in the GBLUP context, where I'm running a bivariate model, and that's the mixed model equation. And below, I depict the solution for the univariate and the univariate conditional to the second variable. So everything that I'm highlighting here is explicitly the additional information that G1 is gaining from being fit together with G2. So you really can analyze and quantify this gain as a function of the various components here. In addition to that, this association between signals does not necessarily have to be dense. So what we have seen that in the slide before is that the generic correlation, all signals are correlated, but you can either ask the machine to infer this association, or if you know beforehand, that can be used as well. So one way is to add some sparsity. So in this case, we are clipping out the G1 G3 correlation. So now G1 is independent of G3 conditional to G2. And another thing that can be done is to add directionality. So once the correlation or covariance structure is asymmetric, you're actually saying that, for instance, here the signal from G3 will help G2, and the signal from G2 will help G1. So G1 will not get anything directly from G3, but through G2 we will get some additional information. So those are graphics, and they are very popular on the multivariate modeling. They're usually, once you start adding sparsity, they fall into the Markov random fields framework. But of course, we don't have to necessarily think of mixed model. Multi-response machinery, here are examples on the left, tree type of multivariate. So on the very far left here is just a depiction of a multivariate random forest, where each tree split has to be justified by decreasing the variance in all the responses. So in this case, every time that you make a split, you're actually making it mindful of all responses at the same time. And on the right, just a depiction of the neural network. And what is very nice about neural network is that working with multiple responses comes very natural to this framework, because you're basically just adding more, so to speak, nodes into the last layer. So there is no additional cost to adding more responses in a multivariate neural network. And the nice thing is that it does behave in a similar fashion as I illustrated on the bivariate GBLUP model. As the correlation between traits or between signals increase, you may expect the predictive ability of your predictions will increase as well. So you do gain power according to the generic correlation, regardless of working on the mixed model framework or other frameworks. And of course, there is the classical mixed model framework. Here I'm just illustrating two possible frameworks. So one thing to note is that in the past few years, there has been a lot of research on speeding up the solution of mixed models and making those models accommodate many more observations and traits. So on the top, I'm illustrating a mega LMM from Dan Runcie from UC Davis. So his proposed framework can fit traits on the thousands. So here we have an example on 20,000 traits. And the computational time is quite linear. So it's a nice framework to accommodate many more traits, although it does have some limitations as you increase the number of observations. On the bottom, it's a ridge regression based on the tilde-hat method of variance components and the Gauss-Seidel method to estimate regression coefficients. So the Gauss-Seidel is an exact iterative method, but tilde-hat is somewhat an approximation of RAML. And a nice thing is, even though it's an approximation, you can see that the computational time that you gain out of that is tremendous. I'm comparing here the tags with ASRAML, so AI RAML from ASRAML, and something that would take in this case 3000 observations in a bivariate model would take AS-REMO something between 1 and 5 hours. It would take no more than a minute or slightly more than a minute for tags. And in the trivariate model, something that would take 8 hours for AS-REMO took slightly over a minute. So what you can see is that it can really speed up the fitting of those genomic models. And furthermore, something that I found very interesting is that the coefficients, the variance components, were very similar to the REMO. So the breeding value accuracy was exactly the same. The heritability was also near identical, was equally unbiased between tags and AR-REMO, and the genetic correlations were equally unbiased. However, it was a little less precise using the approximation, but I suppose that's okay. And one thing you can note is that the computation time for this methodology is very linear, so it increases linearly with the number of observations, with the number of markers, and with the number of traits. And that's a plus, because if you feed a model through AS-REMO or any REMO procedure, usually the increase of one trait implies an increase in a k exponential 7. So it's much larger the increase in time from a regular REMO framework. Alright, let's move to the second part of the presentation, bad learning or avoiding bad learning. So in this section I'm going to talk a little bit about metrics of success, a little bit of something to be conscious on machine learning systems, and adequate validation, and something that you can learn or consider about it. Alright, so one thing that I want to point out is that from the whole machine learning systems, once you're considering to really deploy, the code per se corresponds just to a small component of the whole. A lot of having a robust machine learning system that's operating in a halfway is to get all the components together. And in the context of plant breeding, 3 of those boxes, data verification, data collection, and feature extraction, are very breeding something that the breeders would need some hands-on experience to get it very well. So you can imagine that if you're doing the machine learning system in the plant breeding context, the data verification, the collection, and feature extraction is something that could be really helpful if you get a close look from someone who has experience with the breeding per se and is familiar with the data and can ensure that the results make sense. Yeah, so other things that this study hidden technical depth in machine learning systems brings is some of the checkboxes that you should do and test that help you to be aware of strengths and weakness of your machine learning deployment. And some examples of not being so mindful about it are included in this nice book called Weapons of Math Destruction from Cathy O'Neill, where it really gives you the idea of garbage in, garbage out. And many of the biases and problems that come with the data are not solved by the machine. It really comes into the data cleaning and the correct cooking of the data, so to speak. So any biases projected on the data will be projected on the output. And that's where it really helps to design a very mindful machine. So coming back to the plant breeding, you keep in mind that whenever you're doing your machine learning studies, you should be generally chasing the right signal. So for instance, make an effort and try to imagine that this figure on the right is a breeding pipeline. We usually use machine learning or genomic information in 3 stages or 3 steps. So we use either for the advancement of lines, for recycling, which corresponds to choosing new parents and combinations of crosses, and incorporation of new material or bringing new germplasm. So whenever you are thinking about doing recycling, you probably want to consider chasing the additive genetics, right? So you really care about breeding values when you're thinking recycling, because you really want those genetics that are heritable from a generation to the next. In this case, testing machines like a GBLUP, the Base ABC, and Lasso are very suitable for the task. They can usually be used for advancement as well, as they perform reasonably well for advancement. But if you're just thinking more on the advancement side and product placement, then there are not necessarily restrictions of incorporating other machines that are not necessarily linear. And here I'm giving an example of RKGS, DNN, and random forest, because what you really want is just get as accurate as possible on that G/E component and product placement. And one point that I would like to add is that there is not really a clear machinery that should be used for incorporation, because incorporation really means bringing new germplasm to your germplasm, and it's you're really trying to identify what is in lack. So it's not something that is represented on your estimation sets, on your calibration sets. So it's very interesting. Now, next, I would say that once you know what signal you're going after, there are 3 questions that you may ask to see if you're going to the right direction. The why, what, and how of your validations. So the why is, can you define your problem mathematically? So this one is very easy to get it wrong. For instance, if I'm choosing parents to go to my crossing block, I want to make sure that when I choose the crosses or the parents, I'm not only looking at performance, but I'm also looking at, say, effect population size or some measure of diversity that may ensure my gains in the long term, right? So what is how you're going to measure any sort of gain. So in genomic prediction per se, we are more accustomed to use correlations and accuracy. But depending on which area you are, so for instance, disease risk in humans is usually based on MSP, the prediction error, because you really want to get as close as possible and you don't care about ranking, you care about doing the right prediction. Other metrics may include the Jaccard index, which corresponds to selecting the top X% correctly and might be pertinent for selection in general. And another one that's a little more subjective is success 1 or 0, where you have this not necessarily objective definition of success, but you know when a event has been successful or not, and you're trying to optimize for chances of success. And last but not least is the how you're gonna test. So usually you have to start with questions of whether you have the data, and if you don't, you probably want to use simulation. And are you going to do cross-validation, and what type of data? Do you have the data? And another common question you do if you're working with breeding is, am I doing the validations within family or across family? And how can I design adequately my cross-validation? So here's an example where I have 2 systems within family and across family. And for 2 questions, we have 2 different machines that perform best. So for within family, GBLUP did best, and for across family, deep neural network did best. In this case, it was a public data set, SOINM, where 8 populations were predicting another set of populations. So things to be mindful when you're designing your cross-validation. Here we have the system proposed by Jose Crossa in 2017, where you have layers of data that you have at hand and layers in what you don't have. So for instance, the CV00 would be predicting new genotypes in new environments. Your data set does not have sample of either, so the degree of difficulty is really high. But in case where you either have the genotypes or have the environments, then you're more likely to get higher accuracies. And at the easiest is if you're trying to predict genotypes that you have already observed in environments that you have already observed. That would be the easiest, but probably the least useful. Here's a example that I really like, where I'm analyzing a set of different machineries, BLUP, Base B, Fast Laplace model, RKGS, random forest, XGBoost, and deep neural network, where I train in one set of environments, and I'm validating both on that set of environments, but on unobserved individuals, and unobserved individuals in an untested set of environments. So the first thing that strikes is that whenever I move from a tested environment to untested environment, there is a large leap in predictability. That's, of course, attributed to the GBA component that's not necessarily predictable. Now, within the machines, there shouldn't be too much difference as they are all feeding into the same type of information, as that was a fair cross-validation. But one thing that's really striking is why is the accuracy across family so much higher than within family, then higher than leave a family out, then higher than single family. Basically is the type of information you're extracting. So on a cross family, you have that population structure, LD, and co-segregation or haplotype. Within family, you no longer have the population structure component, so you have a large decrease in predictive ability. So the information changes, you lose predictive ability. Leave a family out, you have population structure because the parents are related to your estimation set, but you do not necessarily have the haplotype or co-segregation because you don't have full SIBs representing the estimation set. On another hand, single family is entirely based on the haplotype effect or co-segregation because your prediction target is full SIBs. They are basically just full SIBs with your estimation set. So you should expect that a cross family would always be larger than within family and leave a family out, and those should be generally larger than the single family, just because of the type of information that you have available and what type of information you can predict or cannot predict, as well as predictions on tested environment will always be better than untested environments. Last, I'm gonna move a little bit more to that ugly side of machine learning, the inner works, a little bit of algorithm. We're gonna talk about optimization and how in machine learning it goes from a ridge regression to a neural network. So first I would start from the discrepancy in philosophy between quantitative genetics and machine learning. The key difference is the optimization function. So basically, in traditional statistics and quantitative genetics, we use the maximum likelihood or restricted maximum likelihood, which is a very complex function. It's not easy to compute, but it's convex, has a unique solution, and it's very well behaved. Usually, you can get to the maximum estimator without much trouble. So the methods we use usually are expectation maximization, MCMC. Also, in second order algorithms include MIVQ and average information, which is particularly used for REMO, right? On another hand, in machine learning, they just care about the regression coefficients, so variance components are not estimated. But the loss function is very simple. So usually they fit that machine just to minimize the mean square error with some penalization like L2. However, because you do not estimate variance components, you incur in having to tune your model to get that best lambda parameter. So you need to do some cross-validation to optimize. That would replace the estimation of variance components. And usually the cross-validation requires a secondary objective function. Usually you do either a simple tenfold cross-validation, but you can do a more elaborate cross-validation to tune your parameters. And then on the traditional machine learning methods, usually the optimization algorithms are based on coordinate and gradient descent, which I'm going to discuss next. So either way, we are always working with the optimization. In this case, is a convex optimization where you really want to reach the blue, which would be the point where all your parameters minimize your objective function. So let's have a look at the example of a ridge regression. So basically, I'm just pointing out the three frameworks that I mentioned above. So coordinate descent is basically you are updating one marker effect at a time and use the residuals to update the next and next and next, and you do that iteratively across the board. So coordinate descent involves building just the diagonals of the left-hand side, which is this x prime x plus lambda. And that's the type of algorithm used for GLMNet, BGLR, BWGR, all those Bayesian methods plus regular ridge regression. Gradient descent, on the other hand, it doesn't build or need the left-hand side whatsoever. You just update all coefficients at the same time. On the other hand, it can be a little challenge because the LD and multicollinearity among markers may make this task a little harder. But the idea is that you just do x prime residuals all the time, and you use that to optimize your beta on the function. So that's the type of optimization that it's all based on matrix multiplication, and that's what you're going to see on your neural networks, in your Keras, PyTorch, MXNet, and H2O. Second order methods involve the preconditioning, or the hashing, as they say, which would be basically building the entire left-hand side of the equation. The great advantage of doing the second order is that it's not an iterative method, meaning you solve this equation, you got the final beta that minimizes this loss function. And that's how most softwares and packages, the general packages are implemented. So ASRemo, LM4, SAS, BluPF90. So they usually are based on this framework. But it does require to build the whole left-hand side equation, which is this side here, x prime x plus lambda. All right. So how does coordinate descent works? So this is well, just the basic derivation of how that solution for the univariate solution came about. But the idea is it is going to update one marker at a time, and the fitness increases over time. As you keep updating your marker effects, you reach eventually the convergence, and your fitness gets better. So that's just an example for a ridge regression BLUP. Now another one will be the gradient descent, also for the ridge regression BLUP. And here's the funny derivation for gradient descent. So an additional parameter that we have to consider when you do gradient descent is the so-called learning rate, is how fast you want to update your coefficients. And that's a problem, again, because of the multicollinearity. If your parameters were all orthogonal, you wouldn't need any learning rate, and a single iteration would suffice. So on the left here, I have a small learning rate where very slowly you achieve the convergence. So here's just a correlation between the final or true coefficient value with the estimated. And took here roughly 35 to 40 iterations to get the right position, the right coefficients. If I use a too large learning rate, it seems that it goes to convergence real quick, but what can happen is what it's called the explosion of the gradient. In this case, it just derails and it gets very ugly. So instead of going towards the right direction, it actually goes to the wrong direction. Finally, I just want to bring how what this solution of ridge regression has to do with the neural net. And for that, I really would say that I don't like this image here. Every time that people talk about neural net, they put this graphic. It's really not informative and does not tell anything about the structure of the neural network. On another hand, if we walk slowly towards the neural networks, that's how it would go. So the linear model is just your design matrix times your coefficients, and you get your fitted values. On the partially square or principal components regression, you have linear eigenvectors. So the way how you'd go for retrieving the fitted values, you multiply your x by the eigenvectors, and then you multiply by the coefficients of the eigenvectors, right? The regression coefficients of the eigenvectors. If you do b1 times b2, you actually retrieve this beta. Otherwise, that's your fitted value. On a single layer neural network, the way how it differs from the partially square in PCR is that you have this activation function that would make your eigenvectors non-linear. So basically, you do xb and you get your eigenvectors. Up to this point they are linear, but then you apply this non-linear function, your activation function, and those non-linear eigenvectors will be multiplied by the regression coefficients beta 2, and then you get your fitted values. In the deep neural network, you're basically just adding more layers. So basically you get your eigenvectors here, you apply the activation function, then you multiply by other set of coefficients, you apply again the activation function, and then in the end you multiply by your regression coefficients to get your fitted values. So the idea is that by having more activation functions, you get more non-linearities to be captured by the neural network. So in the end, really, I mean, the neural network could be very easily framed as a linear system, but for some reason people prefer to use that ridiculous picture. So that's how the so-called scary deep neural network is fit. So the whole code to fit a neural network Here it took me just like roughly 30 lines from top to bottom, including comments. Including in here I'm implementing the dropout and all the workabouts. But everything can be broken down into 3 steps. So basically it's feeding the layers. H here stands for the hidden layer, so to speak. Where is the XB and then the activation transformation. Then you do the same for the second and same for the third. And here H3 is basically your fitted values. Right, your H3 here would be your fitted values. Once you have these values that were started at random, you're going to calculate the gradients, which are like the residuals, right? The fancy name for residuals. So all you do is do y minus, observe minus fitted values. You get your residuals. You do what they call the back propagation. So take those residuals and multiply by bit 3, and then run the activation function. And you run that one more time to back propagate to the first layer. So also, you can notice that everything here is based on matrix multiplication. So one thing that's very nice about the neural network or deep neural network framework is that you're not inverting anything, you're not working with Hessians, you're just doing matrix multiplication. And last, you just run the update of the coefficients for through gradient descent, as I showed in the previous slide. Very simple. You update your coefficients. Usually the learning rate will be very low in the deep neural network in comparison to the one we discussed in the ridge regression, just because you have so many more coefficients to optimize that you really want to run that slowly. But it's very simple. So basically then you're going to update your coefficients and go back to feed the layers, compute gradients, and update coefficients again. And it's further efficient because usually you don't compute the whole data you have at once. You do that in batches, so you sample at random, say 5% or 10% of your data. You run one cycle, and then with your new coefficients, you use another batch to calculate the next cycle. So it actually is computationally very efficient. But there's, of course, no guarantee that if you run twice the same neural network, you're going to get the same degree of predictive ability. But in general, it's a great machine and it has a lot of potential, especially to combine data from various sources. So what at first would look as a very scary method, the deep neural network actually is a rather simple machinery. So at last, just to reiterate, so during the presentation we saw that machine learning is a very powerful tool. It can be applied in various aspects of plant breeding. We went through and discussed a little bit about cross-validations and some considerations we have to have once we are designing the cross-validation system, and how that helps us to understand the machinery and weakness and strengths and what type of information is being captured. And last, even the most scary black box, the deep neural network, can be easily broken down into simpler machines. I hope you have enjoyed this talk, and thank you very much.